Hello everyone, welcome to Smita Kiran Biology classes. I hope you are all good. In this video, let us discuss in detail about DNA replication in prokaryotes. Prokaryotic cell example, the prominent one is Escherichia coli. The replication require a set of catalysts, and those set of catalysts are called enzymes. You have studied that is enzymes are the biological catalyst which will bring about various types of reactions. Among them, the first enzyme involved is the DNA dependent DNA polymerase. It is the main enzyme involved in the process of replication. It uses DNA as a template and synthesizes DNA. Therefore, it has got the name DNA dependent DNA polymerase. Now, the DNA polymerase enzyme is found to be highly effective, that is highly efficient. It is highly efficient because it can polymerize. Polymerization means addition of more number of nucleotides. The polymerization of large number of nucleotides will take place in a very short time. That is, the DNA template, the complementary DNA will be synthesized in a very short time. For example, in Escherichia coli bacteria, the length of DNA is measured 4.6 into 10 to the power of 6 base pairs. That is the length of DNA. To complete the process of replication, that is the complete DNA will be synthesized or replicated within 18 minutes. So, on an average, there is an addition of 2000 nucleotides on a complementary strand per second. So, that is very important. Per second, 2000 nucleotides. So, you could think such a large number of nucleotides are added in a short time in a accurate, so there is not be any mistakes. If at all there are any mistakes, it will leads to the mutations. So that is about the first enzyme involved, that is DNA dependent DNA polymerase. The second enzyme involved is helicase, then followed by topoisomerase, a kind of DNA gyrase enzyme. Along with that, there is a involvement of DNA ligase enzyme. The role of these enzymes I will explain while explaining the process of DNA replication. And another raw material for the process of DNA replication are deoxyribonucleoside triphosphates. They are otherwise referred to as DNTPs. Here it refers to deoxyadenosine triphosphate, deoxythymidine triphosphate, deoxyguanidine triphosphate and deoxycytidine triphosphate. These are the raw materials for the process of DNA replication, and these are found to have serving a dual purpose. So this could be important for 2 marks. They act as a substrate, that is the first purpose set by them. So that is during the polymerization process, DNTPs will be added on a complementary basis to a template strand. So for that they will be acting as a substrate. And secondly, they provide energy for the polymerization of nucleotides, because this process is a highly energy requiring process. Now in DNTPs there are 3 phosphates and there are 2 terminal phosphates present on either side. During polymerization, these phosphates will be broken. So whenever there is a detachment or a break of these terminal phosphates, it will result in a release of energy because they are energy rich compound. So you may ask with the question, which phosphate store the energy like ATP? So that is terminal phosphates, but another phosphate will be added to the DNA. And sixth requirement is oricite or origin of replication. This is the site at which actually the process of DNA replication starts, or replication start points are called the oricites or origin of replication. They are found to be very important during the recombinant DNA technology process. So we will be discussing there also this one. Now let us explain the process of DNA replication in detail. I said in a double helix, there is presence of ori site. There are many ori sites present, like where I am writing here. So there are 3 ori sites. For example, I am telling, not exactly in any organism. Let's just to explain, I am telling here. First, what happens? Helicase enzyme will binds to origin of replication, that is ori site, and it will uncoil the DNA in a very short length. The very short length of DNA will be uncoiled by the breakage of hydrogen bond. This is very, very important, because the DNA replication process will be highly energy requiring process. So whole DNA will not be separated at a time. Okay, so why there is a uncoiling of only small stretch of DNA? Because it is a highly energy requiring process. This uncoiling will results in a small length of DNA which is converted into single strand, and that is called DNA replication, actually in which the process of replication or polymerization of nucleotides will take place. And on either side, the DNA is being double stranded. If you could imagine, if we uncoil a thread, a long thread or a rope at one end, then there is a super coiling observed. Same way it happens in the DNA double helix also. Whenever there is an uncoiling, it will result in super coiling in the DNA double helix. And that super coiling may interrupt the process of replication. That's why the topoisomerase or DNA gyrase enzyme will nick the DNA either in a single strand or in a double strand and releases the stress, and it will rejoin it. Okay, so you have to remember what topoisomerase will do. It will nick the DNA double strand, both the strand or single strand, and releases the stress and rejoin it, so that the DNA replication will undergo uninterrupted. Got it? So now what are all the points clear now? Helicase will unwind the DNA and convert it into single strand, and at a small stretch there is a process of replication taking place, and that is called replication fork. Okay, so if we write separately the replication fork here as it is given in NCERT text, it will be happening in both the side. One side replication fork is given, which consists of parental DNA. Here is parental DNA, I am writing separately, and this parental DNA will be separated by the activity of enzyme, that is helicase, so that it will be converted into a single strand, and that process will take place at ori site, and it will be by the enzyme helicase, and remaining will be the double strand. Now the one strand will be running in 3' to 5' polarity, and the opposite strand will be running in 5' to 3' polarity. Meanwhile, during the uncoiling, there is a creation of stress, and that stress will be released by topoisomerase enzyme. Now let us understand what is exactly the role of DNA polymerase enzyme and how replication will take place in both the strands, that is both the template strands, whether it is the same or the different. Now DNA polymerase enzyme will always polymerize the nucleotides in 5' to 3' direction. They may ask why. Because it required the polymerization by DNA polymerase require the replaceable OH group, and OH group is always available at 3' end. So at that template which is running in 3' to 5' direction, the process of DNA polymerization will take place continuously in 5' to 3' direction. So the process will be faster. That's why this is called leading strand, or it's also referred to as continuous strand. So there is no interruption in between during the synthesis of DNA at the template having the polarity 3' to 5'. Whereas the opposite strand is having some complexity because it is having the polarity 5' to 3'. So here you have to imagine, if the polymerization has to take place in 5' to 3' direction, it has to had the nucleotide at 3' end, but there is no template available here. That is why at the strand which is having the template strand having polarity 5' to 3' end, there is a synthesis of RNA primers at regular intervals. Whenever there is an uncoiling, there is a synthesis of small stretch of RNA. We will call it as 5' RNA primer, and DNA polymerase will add the nucleotide to that length. Okay, so this is one. and the continuation of process, that is whenever the process of uncoiling will continues, another molecule of RNA primer is synthesized, again DNA polymerase will polymerize it, so on, it will goes on. But in the opposite strand, the synthesis will be continuous. But in a template strand having polarity 5' to 3'N, the synthesis is discontinuous because of the synthesis of many RNA primers. That's why the process will be lagging. That's why this strand is called lagging strand or discontinuous strand. So you may ask question, so in opposite strand there is a presence of RNA? No, not at all. These RNA primers which are synthesized are removed after, and these discontinuous strands which are synthesized are called Okazaki fragments. Later, during the completion of DNA synthesis process, that is replication, the DNA strands, that is Okazaki fragments, will be joined by the enzyme DNA ligase. Okay, so that is how the process will continues. Okay, so now for your understanding, here is a DNA which is having two strands, that is 5' to 3', 3' to 5'. And process of DNA replication, where it starts at ORI site. I said there are many ORI sites present. Remember, at all ORI sites, the process of DNA replication will start simultaneously. Don't imagine at one site ORI replication will start and complete and another will begins later. Not at all. Or in all the sites, there is an uncoiling of DNA and synthesis of strand continuously at a time. Okay, so here in one strand, in one direction continuous, another direction discontinuous, in one direction continuous, another direction discontinuous. So the process will happen in all the ORI sites at a time simultaneously in a same rate, so that the process of replication will complete in a very short time. After completion of the process, all the ORI sites will be joined. Whenever there is a joining of all the ORI sites, then there is synthesis of two molecules of DNA which are similar to that of the parental DNA. I hope you are all clear with the DNA replication process. If you have any doubts, please comment in the comment section. I'll come up with the next video. Thank you.